Good afternoon, one and all. Welcome to Shanvi Learning Services. Welcome to our biology session. Please stay online. So you know already what is the reason behind of muscle pain, right? See here, how discussed in last class, lactic acid was found to vary greatly in its concentration, as we see from the graph here. X-axis time in minutes mentioned, and y-axis concentration of lactic acid in blood milligram per centimeter cube. Okay, and there is a chance to ask this type of questions by giving like these graphs in your annual exam. Okay, they'll give particular graph by giving x-axis, y-axis, and x-axis they will plot that time in minutes, and y-axis concentration of lactic acid in blood milligram per centimeter cube. By giving that information, there is a chance to ask some questions on that concept. By observing graph, we have to answer for the given questions. We have to answer for the given questions. So, just to have an idea about that graph. And here, accumulation of lactic acid. Accumulation of lactic acid. When athletes or anybody go for vigorous exercise, then we can observe that pain in muscles. Okay, if you also just do for some time a vigorous exercise, you will get pain in muscles. Why you are getting pain in muscles? Due to formation of lactic acid in your muscle. Due to formation of lactic acid in your muscle. Because of that reason, you are getting pain in your muscle. Accumulation of lactic acid result in muscle pain. Okay. See, take walk like brisk walk or slow jogging or running. Okay. For same distance, we feel that there are increase in pain levels. There are increase in pain levels. This is because of, as I told just now, this is because of ACE, lactic acid accumulation. This is because of lactic acid accumulation. Okay, so accumulation of lactic acid leads to get pain in muscles, right? Then the active muscles and then only grantedly removed from the blood after exercise. Active muscles and see here, pain levels in muscles because of lactic acid accumulation, the active muscles, and then only gradually removed from the blood after exercise. The working muscles was being changed to lactic acid. The working muscles was being changed to lactic acid. The energy stored in lactic acid molecules. The energy stored in lactic acid molecule is less than that in sugar molecules. The energy stored in lactic acid molecules is less than that in sugar molecule. And if the acid comes from the acid if the acid comes from the sugar, then the energy released. Okay, the energy stored in lactic acid molecule is less than in sugar molecule. The energy stored in lactic acid muscles. The energy stored in lactic acid molecules. Lactic acid molecules is less than that in sugar molecules. Okay, repeat once again. The energy stored in lactic acid molecules. Energy stored in lactic acid molecules is less than that in sugar molecules. So when compared to sugar molecules, in lactic acid molecules, energy is less. And if the acid comes from the sugar, then the energy released. Okay. Okay. The energy then the energy is released and could be used to rebuild ATP. Released energy could be used to rebuild ATP, adenosine triphosphate, from ADP and phosphate. Adenosine diphosphate and phosphate. The energy stored in lactic acid molecule is less than that in sugar molecule. And if the acid comes from the sugar, then the energy released and could be used to rebuild ATP, adenosine triphosphate, from adenosine diphosphate, ADP, and phosphates. The energy released. Okay. See here, the energy is during the anaerobic breakdown of glucose. Okay. Energy, the energy released during the anaerobic breakdown of glucose. See here, look at the screen. Could be used to, that released energy could be used to rebuild ATP from adenosine diphosphate and phosphate. The energy released during the anaerobic breakdown of glucose. It is not until afterwards, the utility of paints the oxygen needed in order to remove the lactic acid. In order to remove the lactic acid. Okay. It is not until afterwards the utility of paints the oxygen is needed. The oxygen is needed in order to remove the lactic acid. So that's why when they gone for that vigorous exercise, they will get pain because of accumulation of lactic acid. After that, when they are going for rest, how the oxygen is entering into the blood, then slowly into our body, then slowly this pain will be moved from that muscles. So that released energy could be used to rebuild adenosine triphosphate from adenosine diphosphate and phosphate. The energy released during the anaerobic breakdown of glucose. Anaerobic breakdown of glucose. It is not until afterwards the utility of pains, the oxygen is needed. The oxygen is needed in order to remove the lactic acid. Okay, so slowly after you will take that oxygen, then how the oxygen in order to enter into body, then same in order to the pain will be removed also. Okay. The presence of lactic acid in the blood. Presence of lactic acid in the blood. The presence of lactic acid in the blood is the main cause of muscle fatigue. Which is main cause for muscle fatigue? The presence of lactic acid in the blood. The presence of lactic acid in the blood is the main cause of muscle fatigue. F-A-T-I-G-U-E, fatigue. F-A-T-I-G-U-E, fatigue. Is nothing but extremely tiredness. What is fatigue? Extremely tiredness. The presence of lactic acid in the blood is the main cause of muscle fatigue. Fatigue, F-A-T-I-G-U-E, is nothing but extremely tiredness. Okay? Extremely tiredness. But the body is rested for long enough, the tiredness goes. The presence of lactic acid in the blood is the main cause of muscle fatigue. Fatigue is nothing but extremely tiredness, but the body is rested for long enough. The body is rested for long enough, the tiredness goes. Because slowly the oxygen enters into the blood. It is supplied over all the body. So how the oxygen percentage will increase then slowly. Likewise, that particular pain will goes. So it's nothing but like here, the body is rested for long enough, the tiredness also goes. Okay. And the next anaerobic respiration. As you know already, the oxidation will take place in two stages. In first stage, you have flow chart also. The total is belongs to like oxidation. In this, we can see the two types of stages. In first stage, glucose is converted as like a pyruvic acid 3-carbon compound and energy. This process is called glycolysis. L-Y-S-I-S, glycolysis. Glycolysis. This is the first stage. First step. In second step, however here, pyruvic acid and energy is formed. At that time, if oxygen is present, we'll go for the anaerobic respiration. Carbon dioxide will be produced, energy will be produced, water also. Okay? Carbon dioxide and water will be produced, energy release in this. Example, human beings we can. At that stage, if absence of oxygen or really low amount of oxygen or inadequate of oxygen, we will go for the anaerobic respiration or fermentation. In anaerobic respiration, there is absence of oxygen, inadequate amount of oxygen. We can take two organisms here. One is bacteria, one is yeast. The bacteria is lactobacillus bacteria. If we take as an example, lactic acid plus energy will be released. If we go from next example, that is a yeast. I have told you about the bun and bakery items. Then yeast also can be involved in the anaerobic respiration. Absence of oxygen or inadequate amount of oxygen will be released. Ethanol, okay, will produce ethanol, carbon dioxide, and energy will be released. So what is anaerobic respiration? Which respiration takes place in absence of oxygen or inadequate amount of oxygen. That respiration is called anaerobic respiration. Okay, in aerobic respiration, large amount of energy will be released. In anaerobic respiration, some low amount of energy will be released compared to aerobic. Okay, see here, the living things produce carbon dioxide and give out energy. Living things produce carbon dioxide and give out energy. These processes are caused. These processes are caused by an oxidation process. I have clearly given you like how oxidation will take place in two stages. So according to chemistry, addition of oxygen is oxidation. Okay, addition of oxygen is oxidation. Or with respect to hydrogens, we can say that removal of hydrogens. Okay. The living things produce carbon dioxide and give out energy. These processes are caused by an oxidation process. Okay. Let us find out by doing some experiments. Okay. Don't get confused. These experiments are very, very important. Okay. This is a lab activity. The living things produce carbon dioxide and give out energy. As we discussed just now, these processes are caused by an oxidation process. Let us find out by doing some experiments. Now, here, this is lab activity. Some experiments are with yeast. As you know already, when we are going to do the activity, we have to follow a systematic procedure. We have to write first of all aim, after that apparatus. After that, if you want to write separately apparatus and materials required, you can, like glass materials and forceps, like that instruments comes under apparatus. Leaf or some pills comes under that materials required, materials. Okay, in your higher studies, you have to write separately apparatus and materials required. Please try to know what is the difference in between apparatus and materials required. Okay, next procedure. So you have to write precautions also, very, very important, before procedure or after procedure, your wish. In different books, they will give differently. Okay, you have to follow precautions. After that, procedure. Okay, after procedure, observation. After observation, a result. After result, you have to write your own opinion. If result will be 2 or 3 less, then opinion should be more than that. Okay, you have to follow like this systematic manner. And many of the students don't know, and they will write procedure only one and a half page. By that, you will get just 1 mark or 1.5 mark. What mark is given for that particular procedure? Here, aim will be like you will have offer 1 mark for aim and operator separately and procedure separately and observation, everything. Or sometimes they will mention particular aim, operators, procedure and result. Then that what they mentioned in that question, that compulsory you have to write in your answer sheet. If you miss anyone also, you may have chance to lose your marks. So every heading, if they not mention anything, then every heading will have the priority. If they mention particular things along with that normal sequence, you have to highlight that particular given headings. So here we have lab activity, some experiments with yeast. First of all, aim to prove anaerobic respiration, right? What we are doing here? We are going to prove anaerobic respiration. To prove anaerobic respiration. See here, we need operators, right? Here, I will show one operators. Here we need thermoplast. Okay, look at the screen. On the screen, thermoplast. Next, we need thermometer. We need thermometer. Here, there is a chance to ask some like different. So don't get confusion. There are some different activities are there. Here, testing for production of heat and carbon dioxide under anaerobic respiration, and carbon dioxide, a byproduct of energy release, and like the different and evolved CO2 in respiration. Evolved CO2 in respiration is there, separately is there. Heat evolved during respiration separately is there, and now this is like a testing for production of heat and carbon dioxide under anaerobic respiration. So first you have to understand the question. You have to understand the question, then give your answer. Otherwise, you may have chance to lose here. Here we have 3 activities: carbon dioxide and heat. In one activity, we can have chance to observe heat, testing for production of heat and carbon dioxide under the anaerobic respiration. That is different. And separately, for carbon dioxide, a byproduct of energy release is separate. And here evolving CO2 in respiration, and evolving of heat and evolving of CO2 in during respiration, separate activities are there. Okay, so be careful, concentrate on that. Here, let us come to our lab activity. Some of experiments with yeast. Aim is to prove anaerobic respiration. Here apparatus, thermoplast. Now we are in thermometer on the screen. Like next, test tube. Test tube. Next, yeast. Yeast boiled and cooled glucose. Okay, yeast boiled and yeast, boiled and cooled glucose. Boiled and cooled glucose. Bicarbonate. Bicarbonate solution. Here bicarbonate formula HCO3. HCO3 is a bicarbonate. Bicarbonate solution. Next, here we need liquid paraffin. PARAFFIN. Liquid paraffin. I repeat once again, apparatus required: thermoplast, thermometer, test tube, yeast, boiled and cooled glucose, bicarbonate HCO3 solution, and liquid paraffin. What? Liquid paraffin. Next, procedure. take a glucose solution. Take a glucose solution in a flask. Okay. You have to take a glucose solution in a flask. If glucose solution, how to make glucose solution? If you want, so where glucose is present, you will have many. So just to take a powder and add some amount of water and stir it well, then glucose solution is ready. Okay. In which powder glucose is present, that glucose powder you can collect and add a few drops of water to that collected sample, which is like how many drops of water is suitable for that particular collected sample. You have to collect and you just add some drops of water to that glucose powder and stir it well. Then glucose water is ready. Glucose solution is ready, right? So here take a glucose solution in a flask. It is boiled and cooled without shaking to remove oxygen. See, after taking glucose solution, we have to boil and cool. So don't shake that. Okay. Why we need to boil and cool? To remove oxygen. Because it is very, very important. We are doing some activities on anaerobic respiration. Anaerobic means, you know already, there is absence of oxygen or inadequate amount of oxygen. So here, while you are taking, already oxygen is present in this. We have to remove the oxygen, otherwise we cannot do that anaerobic respiration activities. That's why you need to remove that oxygen by cooling and by boiling and cooling without shaking to remove oxygen. Okay. So take glucose solution. You, before going to do this procedure, you have to get ready with the glucose solution. Okay. You have to prepare glucose solution and be ready in one beaker or any, okay, glass material. Take glucose solution in a flask. It is boiled and cooled without shaking. Why? To remove oxygen. To remove oxygen, because here we are going to do that anaerobic respiration. Activities on anaerobic respiration, that's why we have to remove the oxygen. If oxygen is present in that only, we may not get a proper result, because here we need inadequate oxygen or absence of oxygen. Now put some yeast. Take a glucose solution in a flask. It is boiled and cooled without shaking to remove oxygen. Now put some yeast. Put some yeast. Here, pour some liquid paraffin. Pour some liquid paraffin on the mixture. Look at the screen on the mixture. Why we need to pour liquid paraffin on the mixture? Yes, to cut off the supply of oxygen. If you did not that, then however, through that whole compulsory oxygen will come. That's why here we are doing that anaerobic respiration activities. So we need to see that without oxygen or absence of oxygen or inadequate amount of oxygen. Pour some liquid paraffin on the mixture to cut off the supply of oxygen. Okay. Arrange a wash bottle. Arrange a wash bottle containing a bicarbonate. Arrange a wash bottle containing a bicarbonate, HCO3 bicarbonate. Containing a bicarbonate or indicator solution. Okay. Or indicator solution for the gas produced by the yeast. For the gas produced by the yeast. Repeat. Pour some liquid paraffin on the mixture to cut off the supply of oxygen. Arrange wash bottle containing a bicarbonate or indicate a solution for the gas produced by the yeast. Okay. Now, to check that oxygen, oxygen is removed from the water, to check that oxygen is removed from the mixture, add a few drops of a diagen green solution. Add a few drops of a diagen green solution to the yeast. When you will add this? Before liquid paraffin over it. Before liquid paraffin over it. Okay. See here. To check that oxygen is removed from the mixture, add a few drops of diagen green solution to the yeast before liquid paraffin over it. See here, you have to check out with diagen green solution whether oxygen is present or not. Already we have removed the oxygen by boiling and cooling. Okay, fine. But the oxygen is escaped from that or not, or oxygen is removed from that or not, we have to check. If oxygen is present, we cannot get the proper result because it is anaerobic respiration. Whether oxygen is present or not, we have to check now. So to check that whether oxygen is present or not, we have to add a few drops of diagen green solution. Here we can have a chance to make one question: which solution can be used to check out whether oxygen is present or not? Diagen green solution to check out that oxygen is removed from the mixture or not by boiling and cooling. You have to add a few drops of diagen green solution to the yeast before liquid paraffin over it. Okay. Then how to test whether the oxygen is removed or not? As after this process, this blue dye turns pink when oxygen is in short supply around it. This blue dye turns pink. Okay, this blue dye turns pink when oxygen is in short supply around it. Okay. See here. If you wish to check that the oxygen has been removed from the mixture, you have to add a few drops of diagen green or Janus green B solution to the yeast. Okay, yeast suspension before you pour the liquid paraffin wax over it. Liquid paraffin wax over it. This blue dye means what? Color. This blue dye. Okay. This blue dye turns pink when oxygen is in short supply around it. Okay. When oxygen is short supply around it. When oxygen is short supply around it, this blue color dye turns to pink, such that we can know oxygen is in short supply around it. Okay, here now note down the initial temperature in the thermometer. Note down the initial thermometer in the initial temperature in the thermometer. Here in this flask it is arranged thermometer, and this U-shaped tube is inserted to the other tube. In the tube we have taken like lime water. So you have to note down the initial temperature in the thermometer and observe the mercury level. Observe the mercury level is raised, okay, which indicates the temperature is produced. Which indicates if rising of the mercury, if you observe in the thermometer, which indicates the temperature is produced. Temperature is produced. Then bicarbonate turns a milky white. Bicarbonate SiO3, in that carbon dioxide is present. Bicarbonate turns a milky white, indicates that carbon dioxide is released. When carbon dioxide is released, you know already lime water. What is the nature of lime water? When it's react with carbon dioxide, turns to milky white. This is a universal truth, okay? Lime water is react with carbon dioxide. When the lime water react with carbon dioxide, it turns to milky white. So observe the here we have chance to observe at a time temperature and carbon dioxide, whether temperature produced or not. Okay, and whether carbon dioxide is released or not. So you have to observe the mercury levels is raised, which indicates the temperature is produced, and the bicarbonate turns to milky white, indicates that a carbon dioxide is released. Okay. As time up, I am going to end up the session, and next session let us discuss about the smell and all. Okay. Thank you, Viranda.
Good afternoon, children. Welcome to Sanvi E-Learning Services. This is your physical science class. In the last session, we discussed acids, bases, and salts. So in that, we completed about pH scale. Yes, we discussed some of the topic about pH scale. So yeah, we'll continue the topic. Yes. So pH scale, pH means the power of hydrogen, we know. So we measure the hydrogen concentration in acidic solution, basic solution, and in the neutral solution. So the pH scale gives the information about whether the solution is acidic or basic or neutral, right? So here the strength of acid or base depends on the concentration of H3O plus ions, nothing but H plus ions, or OH minus ions. OH minus ions are bases, right? Produced in solution. So if we take hydrochloric acid hydrochloric acid means HCl, and acetic acid, that is the CH3COOH. Right? So if we take a hydrochloric acid and acetic acid of the same concentration, then they produce different concentration of hydrogen ions. Here, HCl is the acid and acetic acid is also acid. But we observe the different concentration in this. That means the concentration of hydrogen ions is different in these two solutions. The moment of hydrogen ions is more in HCl solution. So we can conclude that this is the strongest acid, and the moment of hydrogen ions is less in acetic acid. So this is a weak acid. So acids that give more H3O plus ions are said to be acids. That means strong acids. And that give fewer H3O plus ions. That means less H3O plus ions are there in the solution, means said to be weak acid. Yes, can you now predict that weak and strong bases are? Yes. So here, if we take the sodium hydroxide and ammonium hydroxide, both are hydroxides only, both are bases, but these strongest bases gives more OH minus ions. The moment of OH minus ions is more in the bases, and the OH minus ion concentration is less in the ammonium hydroxide. Already we perform an activity also based on this, which are strong acids and which are strong bases, which are weak acids and weak bases. So for acids, we'll take the concentration of H3O plus ions. For bases, we'll take the concentration of OH minus ions. By that, we can decide whether the given acid is strongest acid or weak acid, or strong base or weak base. So now, importance of pH in everyday life. So here, are plants and animals pH sensitive? Yes. Here, living organisms can survive only in a narrow range of pH change. So when pH of rainwater is less than 5.6, okay, pH of rainwater is less than 5.6, it is called acid rain. We know in pH scale we have 1 to 0 to 14. 7 is a neutral one. Below 7 acids and above 7 bases. So in this also we have weak acids, strong acids and weak acids. And in bases we have weak bases and strong bases. When we move from 0 to 7, the acidic nature of the solutions decreases. Okay, from 0 to 7, the acidic nature of the solutions decreases. And from 8 to 14, the basic nature increases. Okay, here we have 0 to 0, 1, 2, have a more, that means strong acids. Here 3, 4, 5, 6 are somewhat lesser. That means weak acids. So here, the pH of rainwater is less than 5.6 means it is called acid rain. So when acid rain flows into the rivers, it lowers the pH of the river water. Okay, the pH of the river also decreases. So then the survival of aquatic life in such rivers becomes difficult. Yes, so the aquatic animals will die because the pH values are changed in the river due to this acid rain flows into the river. So here, is the pH change a cause of tooth decay? So here, tooth decay starts when the pH of the mouth is less than 5.5. Okay, tooth decay starts when pH of the mouth is lower than 5.5. So tooth enamel, we know tooth enamel made of calcium phosphate is the hardest substance in the body. It does not dissolve in water. Okay, so this tooth enamel does not dissolve in water, we know that. So that is made of calcium phosphate. So but is corroded when the pH in the mouth is below 5.5. Okay, it does not dissolve in water, but it is corroded when the pH values are decreases. So here the value is below 5.5, then it is corroded in the mouth. Then the tooth decay starts. So bacteria present in the mouth produce acids. Okay, bacteria present in the mouth produce acids by degradation of sugar and food particles remaining in the mouth. Okay, so here the best way to prevent this, to clean the mouth after eating food. Every time after eating the food, we have to clean our mouth neatly. Okay, if the food particles are sugar or food particles are remains in the mouth only, means then the acids, bacteria produces acids, then the tooth decay starts, then the pH of the mouth is reduced to 5.5. So using toothpaste, so which are generally basic. Okay, toothpaste are generally basic, neutralize the excess acid and prevent tooth decay. So toothpaste are basic. So here in our mouth, bacteria produces acid. So when we clean our mouth using toothpaste, so here toothpaste is a base. So acid and base combined together, there it is neutralized. So here we prevent from the tooth decay. So just remember one point here, to prevent this, we have to clean our mouth after eating food. Here, pH in our digestive system. So it is very interesting to note that our stomach produces hydrochloric acid. Okay, in our stomach, hydrochloric acid will produce. So here this hydrochloric acid helps in the digestion of food without harming the stomach. Okay, when the hydrochloric acid produces in the stomach, then only the food digestion will take place. So during indigestion, sometimes indigestion also will happen, right? So during indigestion, the stomach produces too much acid, and this causes pain and irritation. Sometimes we feel that pain in our stomach and heartburns, and sometimes we feel sore taste in our stomach. So that is due to releasing of excess of acid in our stomach. Okay, so at that time we feel indigestion of food. So to get rid of this pain, people use bases called antacids. Right? So these antacids neutralize the excess acid in the stomach. Okay, here antacid is a base. So in our stomach, acid, if excess of acid is produced, means when we take antacid tablet, here acid and base are neutralized. Okay, here the magnesium hydroxide, or we can also call milk of magnesia. So a mild base is often used for this purpose. Okay, this is an antacid. Magnesium hydroxide is a antacid. Yes, you know people that some people will take ENO, right? So you know that very well. At the time of indigestion, people will take ENO to get rid of the pain, right? So that is also a mild base, right? So remember that in our stomach, hydrochloric acid will produce to it helps in the digestion of food. Generally, we know this hydrochloric acid is a very strongest acid, right? So we can't touch that acid with our hands. But in our stomach, without harming, it helps in the digestion of food. Now here, look at this activity 12. First of all, take a diluted HCl in a beaker. Okay, first of all, take diluted HCl in beaker and add 2 to 3 drops of methyl orange indicator. Okay, we need to take in a beaker HCl, diluted HCl. This is not a concentrated. We are taking just diluted HCl. To this HCl, we have to add 2 to 3 drops of methyl orange indicator. This is. So now we have to know the color of the solution. So here, mix a antacid tablet powder to the above solution in the beaker. Now observe the change in color of solution. So here, when we add methyl orange to HCl, the color of the solution red. Okay, the solution color turns to red. So now we are adding some antacid powder. We mix antacid tablet powder to this red color solution. So what is the change in color here? So here, when we add antacid tablet powder to this red color solution, the color of the solution changed into light yellow. Okay, so here, what is the reason for the change in color of the solution? Yes, here, before adding of antacid tablet, the color of the solution is red, right? Here HCl is a acid one. To this acid, we mix the base. So now in the solution, it will form yellow color. That means here acid and base mixed together means we know salt will form, salt and water. So here this is the neutralization reaction. Here this acid and base are neutralized. So here, can you write the chemical equation for this reaction? Yes. So first of all, we are taking HCl. To this HCl solution, we added magnesium hydroxide. Here HCl is the acid and magnesium hydroxide is the base. So what is the product here means magnesium chloride. This is a salt and water. So here, When we balance this chemical equation, we'll take like this here. So 2HCl plus MgOH taken to gives rise to MgCl2 plus 2H2O. This is the chemical equation for this reaction. Okay, right. So here when we add a methyl orange indicator to dilute HCl solution, the color of the methyl orange turns to red. But on adding antacid tablet powder, it slightly becomes yellow. Right? So here the reason we know antacid contains a mild base. Right? So mild base means we'll take sometimes a magnesium hydroxide or aluminum hydroxide. Right? Yes. Next one. pH of the soil. So plants also require a specific pH range for their healthy growth. Okay, plants require a specific pH range for their healthy growth. To find out the pH required for the healthy growth of a plant, we have to collect the soil samples from various places and we have to check the pH in the manner described here given. So like that we have to find out the pH range of the soil. Okay, so we can note down that what type of plants are growing in that region from which we have collected the soil. So here we have to take a 2 grams of soil in a test tube. Okay, in a test tube we have to take 2 grams of soil. To this soil add 5 ml of water. 5 ml water. Now shake the contents of the test tube. Now filter. Here we have to filter the contents and collect the filtrate in a test tube. Yes, this soil and water do not mix completely together. We can observe the soil at the bottom of this test tube. But the water will be changed. Okay, the color of the water changed. We can collect the mud water here. So here, check the pH of this filtrate with the help of universal indicator paper. Yes. Here, when we test this pH of the soil, we can identify the nature of the soil. Okay. So here, what can you conclude about the ideal of soil pH for the growth of plants in your region means? So we can say that fertilizers of acidic nature are to be used in our soil to improve yield of crops in our region. So here, sometimes if the soil is acidic in nature means the farmer would treat the soil of his field with a quicklime. Okay. So when we test the nature of the soil, we can identify that the soil nature, it is acidic or basic, whatever, right? So if the soil is acidic in nature, okay, the farmer would treat the soil of his field with a quicklime, calcium hydroxide, okay, or calcium carbonate, CaCO3, to neutralize the acidic nature of the soil. Okay, here this calcium hydroxide and calcium carbonate are the bases. So if the soil is acidic means when we add this base, it will neutralize. So here, self-defense by animals and plants through chemical warfare. So have you ever been stung by a honeybee? Well, so bee sting leaves an acid. Okay, bee sting leaves an acid which causes pain and irritation. So use of a mild base like a baking soda on the stung area gives relief. Okay, so here, stinging hair of leaves of nettle plant inject methanoic acid or formic acid, causing burning pain. So when the honeybee sting, so it releases methanoic acid. It injects the methanoic acid to the person body. So because of that, we feel pain and burning pain. It is a burning pain. So to that, we have to use a mild base like a baking soda on the stung area. So we feel relief. Okay, so a traditional remedy is rubbing the area with the base natured leaf of dock plant. So which often grows beside the nettle in the field. Now next one, we have salts. Right? So here, in the previous sections, we have studied the formation of salts by the neutralization reaction of base with an acid. So let us understand more about the preparation, properties and uses of salts. Simply we know that when an acid reacts with base, it will form salt and water. So now we are going to learn the preparation, properties and uses of the salts. So look at here, the family of salts. Here, write the formula of the following salts. So here some of the salts are given. We have to write the formula of these salts. So the first one is a potassium sulfate, sodium sulfate, calcium sulfate, magnesium sulfate, copper sulfate, sodium chloride, sodium nitrate, sodium carbonate and ammonium chloride. So here we can write all the formulas here. So potassium sulfate. The formula of the potassium sulfate is a K2SO4. Okay, and copper sulfate. Sorry, here sodium sulfate. Next one is the sodium sulfate. So sodium sulfate is the Na2SO4. And next one we have calcium sulfate. So calcium sulfate, CaSO4. And magnesium sulfate. So magnesium sulfate, MgSO4. Right? And next one we have copper sulfate. So copper sulfate, CuSO4. And next one, sodium chloride. This is NaCl. And sodium nitrate. Sodium nitrate is a NaNO3. And sodium carbonate. This is Na2CO3. And finally, we have ammonium chloride. Ammonium chloride, NH4Cl. Okay, these are the formulas of the following salts. So here given that identify the acids and bases from which the above salts are obtained. So salts having the same positive or negative radicals belong to a family. So same positive and negative radicals. So for example here, sodium chloride, NaCl and Na2SO4. So this NaCl and Na2SO4 belong to the family of sodium salts. Okay, so here this is a sodium chloride and sodium sulfate. So the common name here sodium. So here these are belongs to sodium salts. And similarly NaCl and KCl. NaCl is a sodium chloride and KCl is a potassium chloride. So chloride. So these are belongs to the family of chloride salts. Okay, so how many families can you identify among the salts given above? So here we have the first one is a potassium sulfate. Okay, so this potassium sulfate, when we identify the parent acid and parent base for this potassium sulfate, the parent acid for this potassium sulfate is the sulfuric acid and parent base potassium hydroxide. So why we are saying here parent acid, the formula I'll write here. This potassium sulfate is we know K2SO4. So here given that identify the acids and bases from which of the above salts are obtained. Right? So here the parent acid for this potassium sulfate is a sulfuric acid. Sulfuric acid and parent base potassium hydroxide. Okay. Now the next one sodium sulfate. For this parent acid sulfuric acid. Sodium sulfate. Okay. Na2SO4. Parent acid sulfuric acid and parent base sodium hydroxide. Okay, and next one we have calcium sulfate. So calcium sulfate, parent acid sulfuric acid, parent base calcium carbonate. And next one we have magnesium sulfate, parent acid sulfuric acid, parent base magnesium hydroxide. Okay, and next one we have copper sulfate. Copper sulfate, parent acid sulfur dioxide. Okay, and parent base copper hydroxide. And sodium chloride, NaCl. For this, parent acid hydrochloric acid, HCl, right? And parent base sodium hydroxide and sodium nitrate. Okay, for the sodium nitrate, parent acid nitric acid and parent base sodium hydroxide only. And sodium carbonate, the parent acid carbonic acid and parent base sodium hydroxide. And finally we have ammonium chloride. For this ammonium chloride, the parent acid hydrochloric acid and parent base ammonium hydroxide. So here one more question asked that how many families can you identify among the salts given above? So here we can write the sodium family. Okay, sodium family Na2SO4, NaCl, NaNO3 and Na2CO3. So these are the sodium family. Okay, sodium sulfate, sodium chloride, sodium nitrate, sodium carbonate. All these are the sodium family. And next one we have chloride family, family of chloride salts. So family of chloride salts, sodium chloride and ammonium chloride. Okay, now next one, family of sulfate salts. Sulfate salts are, we have potassium sulfate, okay, sodium sulfate, and we have calcium sulfate and magnesium sulfate, copper sulfate. Okay, these are sulfate salts. And next one, family of carbonate salts. Finally, we have in this family of carbonate salts. Those are carbonate salts. So here, carbonate salts, we can write here sodium carbonate, Na2CO3, and magnesium carbonate, MgCO3, calcium carbonate, CaCO3, etc. Okay, so these are the carbonate salts, right? Now, so next one we have here PHO salts. Now we are going to identify the pH of the salts. So here, collect the samples like sodium chloride, aluminium chloride, copper sulfate, sodium acetate, ammonium chloride, sodium hydrogen carbonate, and sodium carbonate. Now, we have to dissolve them in distilled water. Okay, we have to take one after the other here. First of all, if you are taking sodium chloride, so we have to dissolve that sodium chloride in distilled water. Now we have to check the action of these solutions with litmus papers. Okay, now we have to find the pH using pH paper, that is universal indicator. We have to classify them into acidic, basic or neutral salts. So we have to identify the acid and base used to form the above salts. So we have to record the observations in this table. So the first one given here, sodium chloride. So the sodium chloride, NaCl, okay, NaCl, this is the pH of the sodium chloride 7. This is not acidic, basic. This is a neutral one. Okay, next one we have aluminium chloride. Okay, aluminium chloride, AlCl3. So the pH of the aluminium chloride 7. This is not acidic, basic. This is a neutral one. Now, next one we have copper sulfate. Okay, copper sulfate is the CuSO4. The pH of the copper sulfate is a greater than 7. So here, sorry, here this is below 7. This is the acid one. Okay, this copper sulfate is the sulfuric acid. Okay, this is acidic here. So this is one is a acidic here. This is not basic. and neutral. This one is the acidic here. Now, next one we have sodium acetate. So sodium acetate is the, here sodium acetate is also is one is the base. Okay, like sodium hydroxide, this one is a base here. Sodium acetate, CH3COONA. Sodium acetate is a base. This is not neutral or acidic. Now, next one is the ammonium chloride. Ammonium chloride NH4Cl. Okay, for this, this is one is the acidic like HCl. The parent acid for this is a HCl. Okay, so we can write here acidic like a HCl, ammonium chloride. Okay, this is not basic, neutral, NH4Cl. And next one, sodium carbonate. Okay, here the sodium carbonate. This sodium carbonate is a parent acid is a carbonic acid and a parent base is a sodium hydroxide. Okay, so we'll not take anything here. Just keep the dashes. Now, last one we have sodium hydrogen carbonate. So write the basic for this as a NaHCO3. Okay, it is basic like a sodium bicarbonate. Okay. So here, salts of a strong acid and a strong base are neutral and the pH value is 7. So the salts of a strong acid and weak base are acidic and the pH value is less than 7. Okay, here the salts of a strong acid and strong base are neutral and the pH value is 7. And the salts of a strong acid and weak base, strong acid, weak base we are saying, are acidic in nature and the pH value is less than 7. Okay, and the salts of strong base and weak acid, strong base and weak acid are basic in nature and the pH value is more than 7. Okay, here in such cases, here the pH depends on the relative strength of acid and base. Just here the pH depends on the relative strengths of acid and base. So here further, the formation of salts does not occur as a heat of neutralization is lost to a large extent to ionize the weak acid and weak base. So here in this table, we have strong acids and strong bases. So they are neutral. That means here sodium chloride, the first one given. Here this sodium chloride and ammonium chloride are neutral. That means these are strong acids and strong bases. Yes, and here given one more point. Strong acid and weak base are acidic in nature. Strong acid and weak base are acidic in nature and the pH value is less than 7. So less than 7 means here the copper sulfate. Okay, sorry, the sodium acetate. The sodium acetate and sodium carbonate and sodium hydrogen carbonate. These are less than 7. Okay, so these are acidic in nature. Okay, and finally we have the salts of strong base and weak acid are basic in nature. Strong base and weak acid are basic in nature, right? pH value is more than 7. So the pH value is more than 7 here for copper sulfate. Okay, copper sulfate and ammonium chloride. So these substances, the pH value is more than 7. Yes, okay. We'll discuss the next topic in the next session.